Right, yeah. Okay, thanks so much for having me here. It's a pleasure to see everybody again. And after many years augmented by COVID as well. So it's a real pleasure to be here. I'll just tell you a little bit about what my group has done in thinking about ways to get to early detection of neurodegenerative diseases, because whatever the treatment is, I'm sort of agnostic about that. If you're going to make it effective, you have to try to determine the disease early and get the treatment to people before it progresses too far. So we were always interested in the fact that sensory functions can be affected early in neurodegenerative diseases such as Parkinson's and Alzheimer's. We've heard about the hyposmia mentioned quite a bit here. So for example, olfaction is damaged, according to a study from Hawaii. Olfaction is damaged at least 4 years prior to motor damage in Parkinson's. 90% of Parkinson's patients experience olfactory deficits prior to motor damage. Olfaction and auditory processing are damaged early, perhaps at the mild cognitive impairment level in Alzheimer's disease. And so just as an example, there are markers in neuronal oscillations. This is a study that Patrick is on with Dan Wesson and Raj Kulkarni. And they did this beautiful study where they did micro-injections of alpha-synuclein aggregates, fibrils, into mice. They let them age a few months, and then they did recordings of local field potentials in the olfactory bulb. And they also made sure that there was spreading of the damage. And I just want to show you this picture, which highlights that you can see a tremendous enhancement or significant enhancement of the oscillations at 15 to 34 hertz, the beta oscillations, in these Parkinson's model mice. So that the damage induced by the alpha-synuclein aggregates seems to lead to an enhancement of the power spectrum in the beta band of the olfactory bulb. What that's good for, or why, I'm again agnostic towards. It might be an indicator or a marker that you could use to look for the disease. For Alzheimer's disease, there's something similar, but it's in the gamma part of the spectrum, like 40 to 70 hertz. And these are transgenic mice. This is from the study by Chen et al. Last year. These transgenic mice have significant olfactory deficits relative to wild-type mice. And these APP transgenic mice have a significant increase in the low end of the gamma band power spectrum. They actually have a decrease in the very low frequency oscillations and no changes in the beta band or in the higher frequency gamma band. So you can see that, again, there's an enhancement of the oscillation amplitude in the lower frequency of the gammas, and then there's this, when you average the power in that region, you find a statistically significant enhancement of the power in that region. So there's actually a difference in olfaction between Alzheimer's and Parkinson's. Another thing you could try to go after potentially non-invasively, because that's the other thing I was interested in, is finding things that are non-invasive. Of course, grid cells, which led to the Nobel Prize in 2014 for the Mosers, where you can monitor a single cell in the medial entorhinal cortex as a rat or a mouse is wandering around an enclosure, and it fires on a regular hexagonal grid. There's multiple layers that fire at different spacings. You can actually, with functional magnetic resonance imaging, as was shown several years ago, you can actually detect the six-fold pattern of firing, the six-fold symmetry of firing with functional magnetic resonance imaging. And I just found this study in preparing for this talk by a group from Montreal. It was just published this year. They looked at mice. These are the controls. Y is young, A is adult. These are the transgenic mice, young and adult. This is showing you the six-fold correlation of the grid cell firing in the normal mice. So for the young and adult mice here, you don't see any difference. For the young Alzheimer's transgenic mice, you don't see any difference from these two. But for the older transgenic mice, you lose that six-fold correlation. So this is looking for the red peaks would correspond to where you would see these peaks at 60, 120, et cetera. And you lose those in the adult mice. So I have two students who worked on this, Kendall Berry and Yudo Ji. Yudo graduated and got a job back in China. And Yudo worked on the navigation problem. And Kendall is working on the olfaction problem. how the oscillatory power spectrum changes with damage to the olfactory bulb. And so, just to remind you, the olfactory bulb is located here, close to the nose. That's where the olfactory receptors send things for initial processing. And one observation is that there are oscillations. On top of the respiratory cycle, you see these oscillations, and these oscillations can be in the beta and gamma frequency range especially. Let's see, Lee and Hopfield developed a neural network model. A simple neural network model is a set of nonlinear coupled oscillators that reproduce this type of gamma band activity. So this is not a realistic enough model to include the beta band activity, but does include the gamma band activity. And so Kendall generalized this model to two-dimensional networks, which was lightly touched on by Lee and Hopfield, and scaled it with system size. And then he also said, look, we've got a working network. Let's see what happens if we break it, corresponding to the neurodegenerative damage. And basically, the oscillations come because you have inhibitory granule cells coupled to excitatory mitral cells. The network inputs come to the mitral cells, and they excite the granule cells, which then inhibit the mitral cells. And of course, the mix of excitation and inhibition leads to oscillations. And these are just the equations if you're interested. So then we did 3 different types of damage. We did uniform damage, where we would just uniformly reduce all of the couplings. We did damage that was just in a single column, so localized damage. And we did damage that would spread, more like the spreading we've heard about for alpha-synuclein today. And what happens in the two-dimensional models, if you have both the uniform damage and the spreading damage, you can either damage the inhibitory coupling or the excitatory coupling. You get an increase in the gamma band power spectrum. And the idea is that the decrease in these synaptic coupling strengths could correspond to damage to the synapses, for example, from the loss of microtubules as you get tau aggregation. If you do the localized damage, you just get so the damage strength is horizontal, 100% damage is over here. If you do localized damage, the power spectrum actually just uniformly decreases. But the spreading damage or uniform damage tends to increase the power spectrum, which seems to be at least preliminarily consistent with what's seen in the Alzheimer's disease. And I'll just briefly go over what Udwo did on the navigation. So this is using the continuous attractor model of Barak and Feet, where you have a layer representing one grid cell sheet in the medial entorhinal cortex with a sort of Mexican hat-shaped recurrent connectivity. So there's excitatory connectivity close and inhibitory connectivity outside that. And with this shape to the potential for every one of the neurons in here, you wind up getting the periodic grid cell firing. So if you take a trajectory of a normal mouse and you average it, you'll see the hexagonal pattern coming out from this model. The lowest energy state for the sort of cost function of this is indeed the hexagonal state. And then you can introduce damage, where you parameterize the damage by the radius of a core damage and by the strength of the output weights for each of the neurons in the core, where you multiply the weight function by alpha. And then what happens is you go through a sequence. If you look at the Bragg peaks, you go through a sequence. So you Fourier transform the pattern in real space where the rat is moving, and you get hexagonal peaks for the normal case. You get orthorhombic peaks as you begin to damage it. That goes to sort of stripe peaks, 2 peaks off the central peak as you damage it further. And finally, you get completely disordered system over here. So you can make a kind of phase diagram in this alpha 1/r plane where most of it is hexagonal over here, but then you go through, if you cut any particular alpha to 1/r sequence in here, where r is the radius of the damage region, you can go through this orthorhombic and stripe phase and into the disordered phase. A given trajectory of spreading damage could be modeled by a curve in this plane. The reentrance is because if you globally reduce the coupling, you'll lose the grid cells at a critical alpha value. So it's expected, actually. So the point is there could be non-invasive approaches. You could use electrodes like Iravani et al. Showed. If you could place electrodes on the eyebrows, you can actually pick up the olfactory bulb oscillations. So this shows you, this is the time in seconds and frequency of your signal when you're just in ambient breathing conditions, and this is when you provide an odor. So you can actually see the difference between the odor and non-odor non-invasively with these electrodes attached to the eyebrows. And as I mentioned, functional magnetic resonance imaging can measure the 6-fold firing symmetry of grid cells in the virtual reality environment. That's in Nature Neuroscience in 2018, and this is just showing you the sort of correlations of the positive peaks and anti-correlations with the negative peaks as you go around an angle with the fMRI imaging focused on the medial internal cortex. So there is a potential for non-invasive measurements of these damages well before significant motor or cognitive deficits show up. Auditory localization is another thing you might look at, the cocktail party effect, the ability to discriminate where sounds are coming from declines with age, but it may decline more sharply with Alzheimer's. One problem here is that there's no complete sort of normal state model for vertical sound, particularly for vertical sound localization. So if this is going to be something that could be used with precision, we would actually have to do some normal state science, non-neurodegenerative damage science to figure out how that works. I also just want to say this is personal. My best friend has been diagnosed with Alzheimer's. And one of my best friend's sisters in the late stages of Parkinson's, leading into dementia right now. So in summary, focusing on damage to particular sensory or navigation networks could lead to early detection. Modeling is possible in advance of experiments. We came up with our power spectrum results concomitantly with the data from the Florida group and Patrick. Discrimination between Parkinson's and Alzheimer's might be possible in olfaction, and early detection can lead to potential positive outcomes. For example, I like this one, but I won't push it. I'm not involved. I have no stake in ANLI 138B, but it seems like a pretty interesting drug. So I'll stop there.
So I just have a personal question.
Sure.
Tell me that the cocktail effect as a prodrome to AD is completely speculation on your part. Is that right?
I mean, because it's, yeah.
Because I can't.
I know, I can't either.
My daughter thinks, you're not listening to me, Dad.
Right.
If there's anything else in the room, like someone talking about the Celtics.
Well, it's also, yeah, well, the Warriors actually, but anyway. But actually, in fact, I have the same thing, and I don't hear my daughter as well because my higher frequency detection went out earlier. But
So I do have a question, because I think the olfaction thing is really interesting. Has anyone ever, so as I understand it, they just sent out scratch and smell. There's 8 smells or something, and they get pretty good reading on prodromal PDs. Ken Barrett's work is the one I most. Has anyone actually compared the sensitivity of this detection method versus a smell discrimination test? And then with
right.
versus AD?
Yeah, there's a whole smell discrimination thing. Right, the smell discrimination, of course, gets at if you're at the hyposmia part, right? And this would get at actually potentially detecting changes in the power spectrum of the local field potential. Right. Since this has just been developed, I think the answer is no. But it would, yeah, there might be some, yeah. It's worth looking at, for sure. Yeah.
Seems very hopeful. In your grid cells, what are your 2 real space variables?
They're just literally X and Y. But grid cells has been shown also.
X and Y of what?
For the mouse, it's just X and Y in a, either virtual reality.
I see. Yeah.
But it's also been shown with the grid cells that you can do it. You can do it with head. You can do it with have the head fixed on a screen, and just the eye motion can trigger the grid cell firing. So it does not have to be literally walking around a virtual reality, or it can be. In fact, for that fMRI, it's actually looking at a screen, and it's being cued by moving around the screen with your eyes.
Really great.
Yeah, so maybe you comment a little more about what's actually known about synaptic breakdown in the olfactory bulb, and in other words, your model is very simple, which is great. But what's I mean, maybe there's a question of somebody else, but maybe just a brief.
I would be happy to pull on some expertise. But the one thing I can say that made us feel a little bit more comfortable about this is there are post-mortem examinations of the olfactory bulb, and if you stain for where you see alpha-synuclein deposits,
Sure,
you'll
but that's
see them
but that
show
has the problem of being very late again. So
Sure,
when you're looking at things that are very early on.
It does show up sort of right
yeah,
at the.
right, yeah.
It does show up right at the sort of mitral granule interface.
Well, I'm just
but-
curious whether there's some, I mean, it's not just either on or off. It probably decays in time, and I'm wondering, in the time scale you're interested in, what's known about how fast it decays.
Never really measures you not in mice.
I'm sorry.
Basically, you're saying post-mortem. But if you get
Yeah, no, in the mice, of course, and that's a study that Patrick was involved in. I don't know if you care to comment on it, Patrick, but in the mice, of course, you can follow a time series, and you do see a steady increase in the power spectrum of the beta oscillations.
You see a steady decrease in.
And you also see the spreading of the alpha synuclein also. So you can positively correlate it at least with the measured spreading of the alpha synuclein. It should be said though that's not synaptic. It's all local field potential. So you're not doing patch clamp on an individual neuron. You're sticking the electrodes in the vicinity. Yeah, right.
As far as I know, these beta bands can be picked up in EEG as well in patients. So I just wonder, is that a biomarker that one could use to monitor disease progression, or is that too variable?
That was the hope. That's certainly the hope. Yeah.
Is it being used in clinical trials already or not?
That I don't know actually. That I don't know.